and welcome to Biotecnika. Today we are going to deal with very important thing from Biotecnika we are going to announce. So it's going to be Nail the NEET Revision Series for you. So if you are a NEET aspirant, then this series is for you. So if you want to know about more in detail, you can contact us through the Telegram, which will be given in the description box. And you can also call the toll-free number, which will be also displayed in the screen. And if you want to know more about it, you can also email to info@biotecnica.org. And I would like to inform you that you can also fill up this feedback form, which will be provided in the description box below. After the session gets completed, please do fill the feedback form, which will be given over there. So please confirm if you can hear me and see me. Yes. So I'm just going to wait for some 2 to 3 minutes and then we'll start the session. So I'm going to be teaching you biology. This is Caroline Green from Biotecnika. Just give me 2 minutes and then we'll start the session. Yes, hello everyone. Welcome back to Biotecnika. Okay, if you have any questions on the chapter that I'm going to deal with, you can also put your questions in the chat box. Also, you can also comment in the YouTube. So I will be getting back to you again, okay? Yes. Just give me 2 minutes and then we'll start the session. Yeah, I'm very good. How are you, all of you? Yes. Hello, Harshit. Yes. Okay, fine. So let's start the session. I want all of you to take your NCERT book, your pen, your paper in your hand, because we are going to do a rapid revision session on human physiology. So we have come up with a series for 15 days. And today is the first day. For every day, we'll be coming by 4 to 5 p.m. So you can come every day and check in. We'll be talking about biology and chemistry. Yes. So let's go into the session. Today, I'm going to deal with human physiology. Human physiology usually have the maximum weightage. We can say 18% of marks. In that, I've taken body fluids and circulation. So let's talk about body fluids and circulations. Okay. So we know about human circulatory system. You are going to deal with every line in NCERT. So please take your books, your pen in your hand, and let's mark all the important things, the last minute revision. Yes. Very important thing when we are talking about human circulatory system, there's another important word which is given in your book, which is going to be blood vascular system. This is another important word. Please make sure to underline the point which is given in your NCERT book. I want all of you to do it, which is going to be blood vascular system. So what is this blood vascular system comprises of? Yes, the blood vascular system has blood vessels. It's going to have heart, musculature, heart is given, a branching or network of branching which is given, and even valves are given. So heart, we're going to talk about heart in detail. We'll be talking about blood. We'll be talking about blood vessels. We'll be talking about valves. And again, it's going to be lymphatic system. So first, I'm going to teach about heart, blood, and then we'll talk about blood vessels, heart in detail. So all of you take your pens. Let's start revising. First thing, let's talk about blood. So where is this blood formed? Yes, very important. The formation of blood. That process is called as hematopoiesis, or we can call it hemopoiesis also. Hemato is referring to blood. And poiesis is formation. So blood formation, that process is called as what? Hematopoiesis, very important. So you need to understand where exactly is the blood formed. So when an embryo is there, so usually it will be formed in the embryo sac. So in an embryonic developmental stage, it is in the embryo. So this is important, NEET question. If they ask you where is the RBC or the blood is formed, then you have to remember if they ask embryonic development, you should answer embryo sac. Suppose if the question is asked adult, then you have to mention red bone marrow. This is very important. So after embryonic development, which is formed in the embryo sac or yolk sac, then it will be formed in the liver, and then comes the spleen. Please make a note that spleen is called The graveyard of RBC. Another neat important question. Please make it a star point. I want all of you to do this same. Okay. After that, when you become an adult, then the bone cells are actually formed in the red bone marrow. Where do you find this red bone marrow? You might have studied in your structural organization in animals chapter, where we'll be talking about bones, right? Yes. So where we'll be talking about bones, in this we'll be studying about something called as epiphysis. Epi is something which is above. So this is going to be epiphysis. And in this epiphysis region, you'll be seeing what? Red bone marrow. So make sure if a question comes, in adult, where is the bone blood cells are formed, it is going to be red bone marrow. If it's going to be embryonal developmental stage, it's going to be in the yolk sac. Very important. So we have done 2 questions for your NEET. Next one. Yes. What about blood cells? Yes. So when we are talking about blood cells, blood. Okay, I want somebody to answer me what's the pH of blood. Anybody, can you answer me? What's the pH of the blood? What is going to be the pH of blood? Is it going to be acidic or is it going to be basic? You can just answer if you know. Yes, we know it's going to be what? Slightly alkaline in nature. 7.9 we can say. So it's going to be 7.9 pH. Okay, so blood has 2 components. We are going to talk in detail about it. Plasma. Yes, plasma and formed elements. Where is the plasma actually formed? The plasma is actually formed in the liver. Plasma is formed in the liver, whereas formed elements are actually most predominantly formed in red bone marrow. Just now we have seen in the adult stage. Okay, how much is the composition? Another important NEET question. Yes, 55%. It's going to be 55%, whereas the formed elements, or we can say corpuscles, are going to be 45%. I'll be talking about plasma in detail. We're going to go in for very quick revision of plasma. Then comes formed elements, or we can say corpuscles. Leukocytes. Yes, we know cytes is referring to cells and leuko is referring to white. So WBC. And platelets are going to be thrombocytes. Very important, thrombocytes, which is responsible for clotting of blood. And erythro is referring to red. So it's going to be RBC. So under leukocytes, we'll be studying about granulocytes. How can I remember granulocytes, ma'am? Yes, you can go in for mnemonics. Yes, what's the mnemonic? You can go in for NEB or you can go for BEN. Whichever is easy, you can remember. Granulocytes. Cytes is referring to cell. Granulo is referring to granules. So in these cells, you will see lot of granules. And there are going to be 3 cells, which is going to be, remember, NAB or BEN, which is going to be neutrophils, basophils, and eosinophils. Yes. Then A granulocytes. A is always referring to absence. So absence of granules in the cells. So there are 2 cells, which is going to be lymphocytes and monocytes. Yes. So we have studied about the blood. Now we are moving on to plasma. Yes, plasma. Another important one which you have to remember. This is very important table. I want all of you to please make sure to write like a tabulation, which is for the final minute revision. Yes. So this is going to be straw color. Yes, we already know viscous fluid. Yes, important NCRT line. The next one, what's the composition? We know the plasma is going to have almost 90 to 92% of water. Remaining are going to be only solute, 6 to 8%. So you should remember this one, 9 to 92% is water. Remaining are going to be solute. So if we have to talk about proteins, another important, make it a star point. If you have your NCRT in your book, please underline all the points and remember the function. Fibrinogen, very important. Plasma is going to have fibrinogen, but it is in an inactive form. That's very important. Don't remember like it is active form. It is inactive form. So fibrinogen is an inactive form, which is going to be clotting of blood. Involved in the clotting of blood? Yes. The next important function of another protein. This is important NEET question. Next one, globulins. Yes, globulins. If you're going to talk about globulins, you have to remember one important mnemonics. Please remember it's IG. So you can remember game D, which is going to be IgG, IgA, IgM, IgE, and IgD. So there are 5 immunoglobulins. So you can remember what's the purpose of immunoglobulins, or we can say antibodies. Yes, it's mainly for defense purposes. Defense, or a fight against infection. So fibrinogen is for clotting, and the next one is for fighting against infections. The next one, albumins. Yes, where you see in the eggs, right? This is mainly for osmotic balance. Another important NEET question. osmotic balance. Yes, 6 to 8% of the proteins we have seen. The next one, minerals. Yes. So plasma is going to have a lot of minerals, sodium, potassium, chlorine, bicarbonates, everything are present over there. Next, gases. Yes, gases, where the gases actually goes in the blood, but where it actually goes in the plasma, in the dissolved form. Yes. So oxygen is going to be 33%, whereas carbon dioxide is going to be 7%. This will come in respiration chapter or breathing and exchange of gases. Important question. Next, nutrients. Nutrients in our book, they have given lipids, amino acids, and glucose, which is the major source of energy. You can also include lactic acid. Okay, the next one, plasma can also carry some nitrogenous waste, urea and uric acid. Very important. Where is the urea produced? This is important. Urea is produced in the liver, or we can say cycle takes place in the liver. Whereas it is excreted by which organ? Yes, it is actually excreted by kidneys. Please don't forget it. It's excreted by the kidney, but it has actually been formed in the urea, which is we know about urea cycle. Okay, we are done with plasma. Let's revise everything quickly. Water, and they're going to have a lot of protein, 6 to 7%. Some nitrogenous bases are present, and they also have some nutrients and electrolytes. This is what given in our book. The next important thing, yes. So we will talk about next formed elements. So this is going to be 45%. Yes, RBC. Yes, we already know about RBC, which we call erythro is referring to red, sites is blood. And the formation of RBC is called as erythropoiesis. I told you poiesis is referring to formation. Erythro is referring to formation of blood. Hematopoiesis is formation of blood, but erythropoiesis is formation of RBC. Yes. How much is the total number of RBC? Yes, this is given in our NCERT book. 5 to 5.5 millimeter cubes is present over there. RBC is present. You should remember this one. This is another important question. It has been repeated in the previous year question paper. If you have in your book, please mark it or make a star point over there and write this one, make it a stick point over there. Immature RBC has nucleus, which means when the RBC is actually formed, it has nucleus. It's nucleated, very important. But when a RBC is getting matured, it becomes what? Enucleated, no nucleus. Okay, why there is no nucleus? Let's check. RBC. Suppose if I have to talk about RBC, let's take RBC. RBC is going to have hemoglobin. Yes. What's the role of hemoglobin? Hemoglobin is going to have iron, right? Yes. Or we can say it has Fe in the center. And they're going to carry 4 oxygen molecules. They're going to carry 4 oxygen molecules. So what's the purpose of RBC? RBC is a transporter of oxygen. So here what exactly happens in RBC when they are becoming immature? They're going to lose all the organelles. So no nucleus, no mitochondria, no ER, no Golgi, nothing is present over there. Then how do they perform respiration? Anaerobic respiration, important. They perform because they are best oxygen transporters, but they do not use oxygen for its own purpose. That's very important. Okay, apart from that, so how much? 100 ml. This is important NEET question. Make it a star point. 100 ml of blood is going to carry how many gram of oxygen? Hemoglobin. Important question, NEET exam question. All of you make it a star point. It is going to be 12 to 16 gram of hemoglobin. Please make it a star point in your NCERT book. It's going to be 12 to 16 gram of hemoglobin. 1 gram of hemoglobin. Suppose 1 gram of hemoglobin is going to carry 1.34 ml of oxygen. This is important. It has a correlation with breathing and exchange of gases. Okay, so we are done with this one. So immature RBC has a nucleus, whereas mature RBC has nucleus. Immature RBC do not have nucleus. We already know the shape. What is the shape of the RBC? Everybody knows. The shape of the RBC is going to be like this, biconcave, or it can also be spherical, right? Yes, but we already know about sickle cell anemia, which is going to be sickle cell shape. What's the lifespan of RBC? RBC is going to have a lifespan for almost 120 days. After what happens to this 120 days? They're going to die somewhere else. What's that important question? Which is going to be graveyard of RBC. RBC is going to be destroyed in spleen. Important question. And we can say this also. Spleen is called as the blood bank of RBC also. In our book, they have given graveyard of RBC, as well as we can say blood bank or we can say of RBC, bank of RBC. We'll write bank of RBC. Important. Okay. Suppose if a person is going to have more amount of RBC, what's that condition called as? Many. Poly means many. So polycythemia means maximum amount of RBC. If a person is going to have maximum amount more than 5 to 5.5 million millimeter cubes, then it's going to be polycythemia. Suppose if a person is going to have less amount of RBC, which means hemoglobin has become less. Don't get confused with if hemoglobin is less, that condition is called anemia. If RBC is less, that condition is different. That condition is going to be erythropenia. Erythropenia, let's not write poly. Erythropenia, red blood cells, penia is like pain. So when there's going to be pain, ultimately there's going to be less amount of polycythemia, or we can say erythropenia. So you should remember all these things for RBC. Now we are moving on to WBC. Yes, WBC is otherwise called as leukocytes. Yes, we already know. This process is called as leukopoiesis. Poiesis means what? Formations. Yes, so WBC formation, important question. Yes, so here you have to remember granules, agranular leukocytes. Granules, I told you how to remember this one, Ben or Neb. So you should remember basophils, eosinophils, neutrophils, and then agranulocytes, which do not have granules. Here you can find a lot of granules over here. Here also you'll find a lot of granules, but in this case you'll never find any granules here, which is going to be monocytes and lymphocytes. Lymphocytes are T lymphocytes and B lymphocytes. T cells matures in thymus. Important. T cells matures in thymus. That's why it's called T lymphocytes. B cells matures in red bone marrow only. It is formed in red bone marrow and it matures also in red bone marrow. So you should remember all these things. Remember which is granulocyte, which is agranulocytes. Okay, we already know RBC is going to be 5 to 5.5 million millimeter cubes. So we already know about it. What about WBC? It's going to be 6,000 to 8,000 cells. Important. And what is the shape? It's going to look like amoeba. Yes, it's going to look like amoeba. And very important, I told you, immature RBC has nucleus. Mature RBC do not have nucleus. So we used to take into consideration our RBC is enucleated, or we can say no nucleus. But in WBC, they have nucleus, and that's why we can stain them and we can view it under the microscope. Okay, they have any hemoglobin? No, they do not have hemoglobin. That's why they do not have any pigment. But RBC has hemoglobin. We know for 100 ml, 12 to 16 gram of hemoglobin are present. This is important, guys. This is very important. I want all of you to take a screenshot of this one. This is really important. This is given in our book. So we will talk about granulocytes, agranulocytes. I want some answers from some of you. So can somebody answer me? What are granulocytes? What are granulocytes? So we know granulocytes are going to have some granules, right? They're going to have some granules which has a lot of things over there. So we already know basophils, neutrophils, and then it's going to be eosinophils. So we will make everything granulocytes. The rest monocytes and lymphocytes are going to be agranulocytes. Yes. What is the number? First, we'll take up which is the most abundant one in a WBC. It is going to be neutrophils. 60 to 65% of WBC, they actually occupy. Important. So they can ask you which is the most abundant WBC. So you should remember neutrophils. Yes. So neutrophils are going to be 60 to 65%. And if you see a neutrophil, suppose if I have to draw a neutrophil, neutrophils are going to be multi-lobed nucleus. So the nucleus are going to be many lobes like this. If you see, there would be many lobes like this. Many lobes like this. Suppose if you're talking about this one, it's going to be phagocytosis. They engulf foreign bodies. They engulf all the foreign bodies. And very important, why the names comes neutrophils? Neutro means what? They are neither acidic or neither basic, or we can say they are neither stained with acidic ones or with the basic ones. So acidic and basic also you can stain them. So they are neutrophils. You should remember this one. What's the next one? Basophils, or we can say basophils. So this is also granulocytes, and it's going to be 0.5 to 1%. Remember this one? 0.5 to 1%. And the name itself gives you an idea. Basophils is basic stained dyes. So you can stain them with basic dye. So it is stained with basic dyes. What's the purpose of this one? Basophils are mainly for inflammation purposes. They're going to secrete some substances like histamines, which causes allergy. Serotonin. Serotonins can be neurotransmitters. Important. They are neurotransmitters. Heparin. Heparin is going to be anticoagulant. Important. Anticoagulant. If I have to draw the structure of basophil, how to remember basophil, whether they are bilobed or trilobed? There's just opposite of this one. Don't get confused. B is given, so it's going to be bilobed. No, they are going to be trilobed. Eosinophils is going to be bilobed. So if I have to draw this one, they are going to have trilobe structure. So 3 lobes are going to be present. One over here, one over here, one over here. Next, eosinophils. Again, a granulocytes, we can say, and it's going to be 1 to 2 percentage. 1 to 2 percentage, and the name gives you an idea. Yes, eosino means what? Acidic. Phils means liking. So they love to be stained by acidic dyes. So they are stained with acidic. And remember, eosinophils, important question. Eosinophils is always referring to allergic reactions. If a question comes, which of the following is going to be allergic reaction? It's always going to be eosinophil. If it's inflammation which secrete histamine, serotonin, it's going to be basophils. Next important one, monocytes. Monocytes, if you're going to talk, let me draw this for eosinophils. Eosinophils are bilobed. It looks like some headsets. Yes, 2 things on like that. So it's going to be like 2-lobed nucleus, we can say. Next, monocytes, agranulocytes, 6 to 8 percentage. So you can see neutrophils are more, then it's going to be what? Monocytes. Yes. What's the purpose? Phagocytotic cell which engulf foreign bodies. Suppose if a monocyte is present in the liver, we used to call that as Kupffer cells. Suppose if it is present in the lungs, we used to call it as dust cells. Suppose if it is present in kidney, mesenglial cells. Suppose if it is present in the brain, we used to call it microglial cells. Okay, next important one, lymphocytes. Agranulocytes, they do not have any granules. This used to have a bean-shaped structure, like bean-shaped structure. The next one is going to be 20 to 25% is lymphocytes, and they are formed in the bone marrow. And T lymphocytes, as I already told you, it matures in the thymus. That's why it's called T lymphocytes. Yes. So we have done with RBC, we have done with WBC, and now we are moving on to platelets. Yes. What's the role of platelets? Platelets, in our book, they have also given thrombocytes. So platelets are actually seen in males and thrombocytes are actually seen in all the vertebrates except mammals, we can say. Yes. So what does this word means? Megakaryocytes. Mega is something big. Karyon is nucleus and cytes are going to be cells. Suppose if I talk about this, there was one large cell which is having a big nucleus, and this is going to be megakaryocyte, and they have undergone fragmentations. So they break into a lot of pieces, and every pieces have become what? A platelets or thrombocyte. How much is the platelets can be? We already know about RBC, 5 to 5.5 million, and WBCs are 6,000 to 8,000 we have seen. And in platelets, it's going to be 1.5 to 3.5 lakh per millimeter cubes. Yes, suppose if a person is going to have very less amount of platelets, what exactly happened? Dengue can occur, right? Thrombocytopenia. Yes. That condition is called, I told you, penia is pain. Penia is pain. So thrombocytopenia means less amount of platelets. Sometimes people used to get less than 20,000 to 30,000 also. So remember, where is this platelets formed? From the megakaryocytes by cell fragmentation. And the amount is very important, which is going to be 1.5 to 3.5 lakh millimeter cubes. Yes. Okay, how does the clotting process take place? Important question. They'll ask you prothrombins and fibrinogen. So I'm going to talk about clotting. How does the clotting process actually takes place? The same NCERT one, but I'm going to make a flow chart for you for easy understanding. Yes, how is the clotting process takes place? Suppose if a person is getting any kind of injury in the tissue. Let's suppose take injury in the tissue. So what exactly happens? So thrombocytes, or we can say platelets also. Thrombocytes, or we can say platelets. So this thrombocytes and platelets, which are the components of blood, they are going to produce one protein, plasma protein. One plasma protein they are going to produce. And that plasma protein is called thromboplastin. Thromboplastin. And this thromboplastin is going to secrete one important enzyme, very important enzyme, which is going to be thrombokinase. This is important. Thrombokinases. And this is an enzyme, I told you. An enzyme will always need a substrate. So where is that substrate present? Yes, in the liver. In the liver, there is going to be something called as prothrombin. And this prothrombin, whenever any injury is happening, it will come from liver and it will travel in the blood. They will go in the blood, and this thrombokinase comes and activates this prothrombin. So prothrombin is in inactive form. That's why I told you fibrinogen is also inactive in the plasma. Yes, and this prothrombin is going to be converted into thrombins. And now the thrombin is activated. So NCRT has lines over there, so you have to write like a flow chart. Okay, and this thrombin is going to activate fibrinogen. Fibrinogen, which is in an inactive form, and then it is going to convert them into fibrins. Yes, but first when the fibrins are formed, right, they will be soluble. But whenever you have an injury, you will immediately have a clot immediately, right? If it's going to be soluble, then it's not going to happen. So what exactly happened? This fibrin again will be converted into insoluble fibrins. Insoluble fibrin. For this, an important factor is involved. Last line of an NCRT which talks about calcium. Calcium is a factor number 4. There are 13 factors which are very important for clotting process. So you should remember, whenever there's going to be injury or tissues happening in the tissues, what exactly happened? There's going to be platelets or thrombocytes. They're going to produce a plasma protein, thromboplastin, thrombokinase. Thrombokinase comes and activates thrombin into a prothrombin into thrombin, and thrombin will activate fibrinogen into fibrin. So this is going to be the clotting process. Now we are moving on to something called as blood grouping. Yes, blood grouping is very important. I'm going to tell you another NEET question. Carl Landsteiner. They'll ask you the name of the person. Important NEET question. Please write it in your NCRT book or wherever you're referring. Carl Landsteiner. Yes. First thing you have to understand, two important terminologies, which is going to be antigens and antibodies. So let's talk about it. Antigen. Antigen is also a protein. Antibody is also a protein. Antibody is also a protein. Suppose if I have to talk about antigen in detail. So what does this antigen do? So the blood grouping is based on the presence of antigen. Suppose if I have to talk about blood group. Suppose if I'm A positive, so I'll be having antigen A. So if a person is going to be A group, so they will have antigen A. This is very important. So the blood group is actually decided by the presence of antigen. But the question is, where is the antigen present? The antigen is present on the surface of RBC. This is very important. So antigen is present on surface of RBC. Surface of RBC. An antibody is present in the plasma. Dissolved plasma, we can say. It is present in the dissolved plasma. Okay, so let's suppose take one example and we'll understand. Okay, yes. Suppose if I'm talking about blood group A, which means I am also having blood group A. So I'll be having RBC, which is enucleated because it's going to be the mature RBC. So they do not have nucleus. On the surface of RBC, they're going to have antigen A over there. Okay. In the plasma, this is RBC. The blood is going to have plasma also. Suppose if I'm talking about this is going to be plasma. In plasma, what will they have? Okay, they should not have antibody A. If antigen A and antibody are there, they'll react with each other and ultimately what happens? They'll die. So what should not be present? Antibody A should not be present. So you need to have what? Antibody B only. Okay, so we'll write that also. Just give me a moment. Yes. So we'll write, it's going to have anti-B. So I'm A group, so I'm going to have antigen A in my RBC or on the surface of the RBC, but in my plasma, I will have anti-B. Okay, let's suppose take B group. So B group is going to have antigen B. Where is this antigen B present? It is present in the surface of the RBC and they are going to have anti-A. Just opposite will be present. That is what given in our book. So I hope everybody understood. Suppose if I have to talk about AB blood group. Okay, suppose if I have to talk about AB blood group, let me write it over here. For AB group, so they will have both the antigens in the surface. They will have antigen A also. They will have antigen B also. But do they have any antibody? Never. They should never have an antibody. So they have no antibody. They have no antibody. Okay, let's suppose talk about O group. O group. O group do not have any antigen on the surface, but they will have anti-A also. They will have anti-B also. So I'm going to talk about who is the universal donor, who is going to be the universal recipient. Yes, okay. Let's talk about that. Okay, let's write it. So suppose if I'm talking about A group, let's write for B group, let's write for AB group, let's write for O group too. Okay, so where is the antigen present? Antigen is in the surface of RBC. And where is this antibody present? It is present in the plasma. Yes, it's present in the plasma. So we know an A group will always have antigen A on the surface of RBC, not in the RBC, and they are going to have anti-B. And suppose if I have to talk about B group is going to have what? Antigen B on the surface, they will have anti-A. And AB is going to have both antigen A and antigen B. And no antibody, important. They do not have any antibody. That's the reason AB is going to have what? They are universal. Please make sure to make this a star point. It is universal recipient. Because if they have no antibody, they can get antigen from anybody. So that's why they are universal recipients. To be very specific, AB positive are going to be universal recipient. And what about O group? No antigen. No antigen, but they have anti-A also, antibody, and they have anti-B also. So if a person is not having any antigen, suppose if I'm giving O group to anybody else, so what exactly happened? I'm not having any antigen on my surface. So the person will not produce any kind of antibody again. So that's why O group are called as what? Universal donors. To be very specific, O negative is going to be universal donor. So I hope everybody understood this one. So just remember, the blood grouping is based on the presence of antigen actually present over there. The next one is going to be RH compatibility, incompatibility. This is a very important question, I'm telling you, all of you. Erythroblastis fetalis. So let's talk about erythroblastis fetalis. Okay, let's take a condition. So I'm going to talk about first pregnancy and second pregnancy. Before going in for this, we have to talk about something called as RH. What is RH? RH is going to be rhesus monkey. Okay? Suppose I told you I'm going to be A positive. So if I'm going to be A positive, let me take me as an example, A positive. So I'll be having antigen A. And what is this positive referring to? I'm having RH antigen. So this positive is referring to RH antigen. So if I have to talk about A positive person, they'll be having antigen A and they'll also be having RH antigen. Please remember carefully, whoever may be A or B or AB or whatever it is, naturally we do not have antibody against RH. So no anti-RH, or we can say no anti-D. D in naturally in our body. We don't have such kind of thing naturally in our body. Okay, so we are going to talk about what is this erythroblastis fetalis. So let's talk about, suppose let's take the father is going to be RH positive. The father is RH positive. And the mother is going to be RH negative. Mother is RH negative. Okay, if the father is RH positive, let's suppose take the father is RH positive, which means he has RH antigen present on his surface. RH antigen present on it, but this woman is not having RH antigen. So what exactly happens? Suppose if they mate and they're going to have a fetus formed. So if the fetus is suspected to have RH positive, which means they are going to have RH antigen. First pregnancy, what complication it can create? That's going to be this one. So understand very clearly. This is very important. I'm going to consider this is the mother. Just understand this is the mother's womb. Mother's womb, or you can say uterus, whatever it is. And the fetus is actually present over there. Let's suppose take this is the fetus. This is the fetus. And the mother and the fetus are usually connected by placenta. Yes, so this is going to be placenta. Let's suppose take this is the placenta. And this is the mother's blood. So this part is going to be the mother's blood. And this part is going to be the fetal blood. The fetal blood is going to have RBC. Yes, let's suppose take this is RBC. And in this, they are going to have what? RH antigen. So they are going to have RH antigen. The mother naturally do not have any kind of antigen. So no anti-RH, nothing is produced. But what exactly happens when the parturition takes place or during delivery? What exactly can happen? During delivery, what will happen is, you can see over here, just a minute. So you can see, during parturition, when they are going to cut the placenta, or we can say when they're going to cut the umbilical cord, what can happen is the maternal blood and the baby's blood or the fetal blood will mix together. So what exactly happened? Suppose I already told you this is the fetus, and when they're going to cut this one, so here Rh antigen is present in the baby, or we can say fetus. So what can happen is this Rh, when they are going to mix it, so this Rh can travel to the mother's blood. And the mother will think something is coming into my body, which is an antigen. So I have to produce something against it. Naturally, they'll not have. But that situation, the mother starts to produce anti-Rh. So what exactly happened? During parturitions, the mother produces anti-Rh against the Rh antigen of the baby or the fetus. Rh antigen of the fetus. So the mother produces. So this is for only short period of time. The blood just goes and then the mother produces anti-Rh. So what exactly happened? This anti-Rh will travel to the baby's blood, goes to the fetal blood. And in the fetal blood, they already have Rh antigens. So there, Rh antigen and anti-Rh will react with each other. And now what exactly happened? Antigen-antibody reaction. So RBC is going to break and the hemoglobin is going to break. If RBC breaks, bilirubin, biliverdin, everything will come out. The baby is going to suffer from jaundice. And hemoglobin also will break. So what exactly happened? Anemia will also be there. But it's not life-threatening, we can say. It's not going to be life-threatening. So that's why what they'll do some remedies in the first pregnancy itself. So we understood what happens in the first case. Okay, now the mother's blood. Now, what happens in the mother's blood? Now she has produced anti-Rh. Still the anti-Rh is present. Naturally, she is not producing, but during first pregnancy, because of the first baby, she has produced what? Anti-Rh. Now, still it is present in the blood. Now, what exactly happened in the next scenario? Yes, in the second pregnancy, what exactly happened is, since the mother is going to have anti-Rh naturally, so she will be trying to produce this one against the baby's one. So, let's talk about during second pregnancy, what will happen? Okay, let's take during second pregnancy, the mother is again Rh negative, and the baby, this is mother, and the father is Rh positive. Father is Rh positive. The baby is going to be, again, the fetus is going to be Rh positive. So what exactly happens in this scenario? Let's suppose take this is the maternal blood, mother's blood. Now the mother's blood is going to have anti-Rh. The baby is still present here. This is the fetus. And the fetus is connected to them by a placenta. This is by a placenta. And now what exactly happens is this anti-Rh is going to behave like a one important immunoglobulin, which is going to be IgG. And IgG immunoglobulin is the one which can cross placenta even in the embryonal stage. So it can cross placenta. Usually no other immunoglobulins crosses except nutrients used to pass this area. But this anti-Rh can pass through this. So even before a baby is fully formed, this baby is going to have a lot of RBC. In that RBC, Rh antigen is present. So what exactly will happen is this anti-Rh can pass through this and they will react with here. So this antigen and antibody reaction will take place. Ultimately, the baby is going to die. So that's why we call this condition as what? Erythroblastis. This is life-threatening. In the first pregnancy, it's not problem. Erythroblastis fetalis, death of the fetus in the embryonal stage because of the breaking down of RBC. So what we have to do? So in the first pregnancy itself, the mother naturally should not produce any anti-Rh. So what the doctors will do during that case, if they suspect to have that, they will give something called as, or we can say rogum. Rogum. This is a synthetic anti-Rh. This is synthetic anti-Rh. So if it is given to the mother for a short period of time, so the baby's blood is going to mix for only short period of time in the first pregnancy itself. So they will just react with each other, no other complication, because we are inhibiting the mother to naturally produce anti-Rh. So this is going to be erythroblastis fetalis. Very important one, which you have to remember. Next, we are moving on to circulatory system. Yes, very important. So we'll talk about circulatory system. There are two types of circulatory system. One is going to be open circulatory system. Second one is going to be closed circulatory system. Very easy. So do we have open circulatory system or closed circulatory system? Yes, of course we have closed circulatory system. We human beings have closed circulatory system. So let's understand What is open circulatory system and closed circulatory system? Suppose I'm going to make it very simple for you guys. So listen, suppose if I'm talking about open circulatory system. So here what's going to happen? It will be seen in all the low vertebrates. Okay, suppose. Let's take. So let's take, this is going to be one organ, any organ, okay, any organ. So there's going to be blood vessels. So let's suppose take, this is the blood vessels. This is the blood vessel. So what exactly is happening in this case? So the blood actually comes here, and then there is no capillaries. Important. In open circulatory system, there is no small capillaries. No capillaries. So what exactly happens is the blood will be directly poured in the open space. The blood will come out of the blood vessels and it will be poured in the open spaces. And there is a direct contact between the blood and the organ or tissue. Direct contact. So no capillaries, direct contact between the tissue and the blood. This is open circulatory system. Very important. It has a correlation with animal kingdom. How can you remember? Open circulatory system, you should remember one mnemonic. Yes. And please remember this one. Open circulatory system, you have to remember this. Arthropoda. Arthropoda is going to have open circulatory system. Please remember this mnemonic. And non-cephalopod mollusks. Non-cephalopod mollusks. Non-cephalopod mollusks. I want all of you to write it. Mollusks. And the next one, T, is referring to tunicata. Yes, tunicata. And then hemichordata and echinodermata. Starfishes. Yes, starfishes. So all this thing. So in our book, they have given two examples, which is going to be, of course, arthropoda, and we know very specifically it's going to be non-cephalopod mollusks. You have to remember arthropoda, non-cephalopod mollusks, tunicata, hemichordata, echinodermata are going to be open circulatory system. What about closed circulatory system? Closed circulatory system is very easy. You can remember very easily. Closed circulatory system, if I have to talk about. Suppose, let's consider this is going to be the blood vessels. Okay, yes. So the blood vessels usually going to be very tiny blood vessels used to come like this. So now this is called as what? Capillaries. So this is going to be what? Capillaries. Previously, I told an open circulatory system, they do not have capillaries, but here you're seeing a capillary. Suppose this is going to be an organ or tissue. So the exchange of blood is taking place with the help of capillaries. There is no direct contact of blood and the tissue. Here it is going to be with the help of capillaries. So that is going to be closed circulatory system, which is very precisely the most well advanced thing that you can see in all the other cases. The rest of the things after you can see ANTHE. One important example which is given in our book, you should remember, it's going to be Annelida. Annelida is going to have closed circulatory system, and even chordata. We all come under chordata. We are going to have closed circulatory system. Very good, all of you. Okay. The next one we are going to talk. This is an important match the following question. Important question for NEET examination. Okay. Suppose if I have to talk for fishes. So fishes are going to have two-chambered heart. All of you know it's going to be two-chambered heart. And please remember, I'm telling you one thing. Whenever you see in your NCERT book, every line is important. So you might have seen in human circulatory system, the apex is given at the bottom, but apex means tip. But why it is given so? We know in fishes, they are going to have what? Ventricles above and auricles below, right? But as the evolution has taken place, this ventricle has gone down. Okay, so that's why when we draw our heart, we'll draw two ventricles at the bottom and auricles at the top. So ventricle, one ventricle and one auricle is present. What about amphibians and reptiles? Amphibians and reptiles are going to have three-chambered heart, except crocodile. This is important. Crocodiles are going to have four-chambered heart. So we know amphibians and reptiles are going to have three-chambered heart, which means two atria, one ventricles. And in case of crocodiles, birds and mammals, we all come under four-chambered heart. So fishes is going to be two-chambered, amphibians and reptiles except crocodiles are going to be three-chambered, and crocodiles and human beings, or we can say birds, are going to be four-chambered heart. Important, match the following question. Okay. Next one. Okay, we already know about this one. So single circulatory system, double circulatory system, incomplete circulatory system. So if we have to talk about single circulatory system, what does it mean? Listen carefully. This is important, guys. This is very important. Make it a star point. I'm going to talk about fishes. So I'm going to draw the same over here. Just imagine there's gills. In fishes, they have gills. So gills are mainly responsible for exchange of oxygen and carbon dioxide, right? Yes. So they take in oxygen and they exchange carbon dioxide. So this gills is going to bring oxygenated blood. Yes, oxygenated blood. And this oxygenated blood usually comes in aorta. It comes in aorta, and then it will be given to the tissues of the fishes. And now what the blood tissues will do, they'll utilize all the oxygen for its own metabolism, utilize all the glucose, makes all the ATPs for its energy. And now after utilizing everything, they're going to produce deoxygenated blood. Listen carefully. Deoxygenated blood are going to be what? Venous heart. Venous heart. Veins usually carries, very important, systemic veins. I'm talking about systemic veins. Systemic veins carries deoxygenated blood. Systemic arteries carry oxygenated blood, except pulmonary arteries. Pulmonary arteries, important, please make it a star point, all of you. Pulmonary arteries carries deoxygenated blood. Pulmonary veins are going to carry oxygenated blood. Very important. This is an important difference where they'll ask you in your NEET examination. So systemic arteries carries oxygenated blood, but pulmonary arteries carries deoxygenated blood. Yes, now what exactly happened? It comes to auricles and then to ventricles. So which means the heart is going to receive only deoxygenated blood. That's why we call this heart as what? Venous heart. Venous heart. So the heart is going to take this deoxygenated blood and gives back again to the gills. So this is going to be only one circulation that you're going to see in case of fishes. Okay. What about in case of amphibians and reptiles? Yes. So very simple. Suppose if I have to talk about amphibians, heart. Yes. Let's draw it. So this is going to have two auricles. So here one, here one. So let's write right auricles, left auricles, and only one ventricle. So what exactly can happen? So you have to remember this. How can you remember? The right atrium is going to bring what kind of blood? Remember doctor. Yes, everybody wants to become a doctor. So remember doctor. So right atrium will always receive deoxygenated blood. Yes. So right atrium receive deoxygenated blood. So deoxygenated blood comes to the right atrium. Then it will be going over here. And oxygenated blood comes in the left atrium, goes to the ventricle. Now, oxygenated blood and deoxygenated blood, both are there. They will mix with each other. So that's why we call it as what? Incomplete double circulation, where there is going to be mixing of blood that you see in reptiles as well as in case of amphibians, except crocodiles. Yes. The next one, double circulation. So this is going to be our heart. So we know one auricle, right atrium, left atrium, and right atrium, right ventricle, left ventricle. We already know there's a separation, right? Systemic circulation, and we know there's going to be pulmonary circulation. I'm going to talk about this in detail later. So now we will move on to the very important thing, which is going to be the arteries, veins, and we'll be also talking about capillaries. So I'm going to make it very simple for you. All of you can make a note of this one. Arteries, veins, and then we will write capillaries. Okay. So we know there are going to be three layers. One is going to be tunica externa, yes, or adventitia. So remember, E is referring to external, outside. So this is the outermost layer. And tunica media, middle. So it is the middle layer. And tunica intima is the innermost layer. So tunica externa, tunica media, and tunica intima. So this externa, right? Yes, it is made up of fibrous connective tissues. Very simple. Collagen, fiber. And media means muscles. Remember muscles. They are made up of muscles and elastin. The first one is made up of collagen. The second one is made up of elastins. The last one is going to be epithelium. Simple squamous endothelium. So they are made up of simple squamous endothelium. So three layers actually make some important one. But you have to remember what's the difference between arteries, veins, and then capillaries. So let's write arteries. In arteries, what exactly happens? So remember, the outermost one is tunica externa or adventitia externa, right? Yes. And this layer is thinner. This is very important. All of you, please make a note of this. This is thinner. And the next layer is going to be, let me make it this way. So this is going to be Or let me make some other color. Okay, no problem. So this is going to be tunica media. This is thick in arteries. And the innermost layer, suppose if I have to talk about the innermost layer. So this is going to be the innermost layer. And this innermost layer is also thicker, which is going to be tunica intima. This is also thicker. So what exactly happens thicker? What exactly happened to this lumen? Lumen is narrower, very important. So they can ask you a question, arteries are going to have narrow lumen or veins are going to have narrow lumen. So you should remember arteries are going to have narrow lumen, and very important, the outermost layer is going to be thinner, the innermost layer and the middle layers are going to be thicker. The same applies for veins, but with little bit of modification. That's what is given in our book. Veins are also going to have, but listen carefully, veins are going to have the outermost layer is thicker. Tunica externa is thick. And tunica media is also thick. But the tunica intima, right? The innermost layer is very thin. This is tunica intima. So let's write this is tunica intima. This is also going to be thin. Just the opposite. Here you're seeing the first one is thin. In this case, you're seeing the last one alone is thin. So what exactly happened? Wide lumen. So veins are going to have wider lumens, whereas arteries are going to have what? And we know systemic arteries carries oxygenated blood, but pulmonary arteries carries deoxygenated blood. This you have to remember. Capillaries used to have only one layers. Only squamous endothelium is present. Apart from that, you don't have to remember all this thing. Okay. Next, we are moving on to the most important thing of this chapter is going to be human heart. So let's draw everything together and we will understand it. Okay, yes. So let's talk about the human heart. Yes. So let's draw. Okay, let's draw all the atrium and the ventricles. Okay, let's draw it. Okay, let's write. This is right atrium. And I told you, all of you want to become a doctor. So remember doctor. Yes. So doctor. So right atrium will receive what kind of blood? Deoxygenated blood. Very easy. So they are going to receive deoxygenated blood. I'm going to make some changes. So let's write this as left atrium. And this is going to be right ventricles. And this is going to be left ventricles. If you are going to do everything together with me, it's going to be very easy. Okay. So right atrium, right ventricles and left ventricles. Okay. So I told you the right atrium is going to receive deoxygenated blood. But how will they receive? Then have to be some veins. I told you systemic veins. Suppose let me write systemic veins. Just a minute. Okay. Let's write systemic veins. Veins carry deoxygenated blood. And systemic arteries going to carry oxygenated blood. Pulmonary arteries, very important guys, please make sure that you remember everything. Pulmonary arteries are going to carry deoxygenated blood, and pulmonary veins are going to carry oxygenated blood. Important. Okay, let's write everything together. Yes. So the deoxygenated blood actually comes to the right atrium. But how? The upper part of the body has to bring some deoxygenated blood. Who is going to bring it? Superior vena cava. Yes, we will be talking about superior vena cava. So we'll make superior vena cava. So this is going to be what? The superior vena cava. And the lower part of the body also we need to bring all the deoxygenated blood, because the body has utilized all the oxygen. That is brought about by inferior vena cava. So this is going to be inferior vena cava. Okay. Even the heart has tissues. Yes, even heart has tissues. They also might have utilized oxygen. So they also bring some deoxygenated blood, and this is called coronary sinuses. Coronary sinuses. So all these things are going to bring what? Deoxygenated blood to the right atrium. So they are going to bring all the deoxygenated blood to the right atrium. And there is a wall which is actually present here, right? There's an important thing which is present over here. This part. What is this part called as? It is separating the right atrium and the left atrium. This is called interauricular septum. Septum is nothing but the separation. Septum is going to be the interauricular separatum. Very important line in INCERT. It is given. It is going to be the thin muscular. Important. It is not thick muscular. Please, guys, make it a point. It is star point. A thin muscular interauricular septum is actually present between right and left one. Okay. Suppose if I have to talk about this one. Yes. What about this one? What about this one? This is called interventricular septum. Interventricular septum. And this interventricular septum is going to be thicker. This is going to be thicker. This is going to be thinner and this is going to be thicker. So remember, this is thick muscular. Thick muscular. This is an important one. They can ask you which is going to be thinner and thicker, which seems very simple. Every word is important. So this is going to be thicker ones. Okay. What about the auricles and ventricles ones? Yes. This area, this portion and this portion. This is called as what? AV, right AV walls. Very important. You can remember like this. Yes, I'll write it here. Lab rat. Yes, lab rat. Yes. What is that lab rat? It's a mnemonic. You should remember. So left, the atrium is going to have bicuspid wall. So this is the left atrium. So this is going to be what? Bicuspid wall. Bicuspid valve. Or we can call it mitral valve. Mitral valve. Or you can call it left AV wall. Actually, they are going to have a separation, and in that they are going to have opening. In that opening, they have a valve. And this wall is called left AV wall. Yes. So this is going to be rat I've written, which is right AV, which is going to be tricuspid. Yes, you have to remember tricuspid. This is going to be tricuspid. Tricuspid wall. Very important. So this is important. Now we are going to talk about, now the deoxygenated blood has come from superior vena cava and inferior vena cava, coronary sinus, and they are present in this area. Now this will open and the deoxygenated blood will come here. Okay. What exactly happens after this one? Yes. Suppose if I have to talk about this one. Now the deoxygenated blood is here. Who's going to carry this deoxygenated blood? Yes, somebody has to carry it, right? Who's going to carry it to the lungs? Sorry, liver. Okay, yes. So who's going to carry it over there? Yes. So they are going to carry it by pulmonary arteries. Okay, pulmonary arteries. Pulmonary arteries carries this one because deoxygenated blood. Okay, now what is the wall that is present in pulmonary arteries? This wall is called semilunar walls. Semilunar walls. And this semilunar wall is also tricuspid based on how many cusps they are going to have. So it is going to be 3 cusp. So they're going to have 3 cusp, which is going to be tricuspid. And if we have to talk about this one, here oxygenated blood, who is going to bring? Pulmonary veins. There are 2 pulmonary veins which comes from one lobe. 2 pulmonary veins comes from this one. So this is pulmonary veins. They carry oxygenated blood and the oxygenated blood will go over here. And from here, it will be carried to aorta. This is aorta. And aorta is also going to have what? Semilunar walls. Semilunar walls. Okay. Okay, now we have seen that there's going to be superior vena cava, which brings deoxygenated blood. Inferior vena cava brings deoxygenated blood. Coronary sinus also brings deoxygenated blood. And they are poured into the right ventricles. And then this deoxygenated blood is carried by pulmonary arteries and it's given for, or we can say for oxygenation process. And they have been carried again by pulmonary veins to the left atrium and it will be given to the left ventricle. And then it will be taken back again through aorta to all the body parts. Now I'm going to write very important walls that you have to remember, and then we'll go in for questions. Yes. Okay. So first let's write superior vena cava. Okay, superior vena cava. If I have to talk about, do they have any walls? No. Superior vena cava, I told you it is present over there. So they do not have any walls because they are in that angle, so they don't need any walls. No walls. No walls are present. If we have to talk about inferior vena cava, yes, inferior vena cava, do they have any walls? Yes, inferior vena cava has walls, and that wall is called wall of Eustachian. Eustachian. And this wall is actually present here. And I told you there is coronary sinus. This is coronary sinus. So coronary sinus is going to have Wall of sebaceous. Wall of sebaceous. And then there's going to be left atrium, and left atrium has pulmonary veins. And these pulmonary veins, if I have to talk about all the pulmonary veins, do they have walls? No walls. No walls are present. And we said semilunar walls. Semilunar walls. Semilunar walls are present in, we can say it is present in pulmonary artery also, and it is present in aorta also. And we talk about the rest of the walls in detail. So I hope everybody understood the internal structure of human heart. So I'm going to take you to the questions, and then we will see the rest of the things in the previous coming classes. Okay, yes. So let's have some questions for today. Okay, so let's do the stroke volume alone, and then we will just move on to the next one. Yes, this is important. So we know that the heartbeat is 72 times per minute. And we can say stroke volume. Stroke volume. Stroke volume is how much volume of blood is pumped by each ventricle. This is important line. Each ventricle. Every ventricle is going to pump blood. If they ask you both the ventricles, then you answer accordingly. It's going to be 70 ml. So cardiac output, how can we do cardiac output? Cardiac output is equal to stroke volume into heartbeat. So 72 into 70, which almost comes around 5040 ml. So we can approximately take it 5000 ml or 5 liters. So this is one of the things that will definitely get in your examination. So now let's move on to some questions, and then we'll end the session. Okay. So we'll move on to some questions. Yes. Okay. These are all important questions. Which of the following? Yes. And very important. Plasma we have seen. And we will see the rest of the things also. Okay. Which one of the following is involved in coagulation of blood? In coagulation of blood, if we have to talk about, the coagulation is actually done by fibrinogen. Yes, this is the correct answer for this one. Suppose the next one, serum differs from blood in lacking. Which of these things? Serum, very important. Serum, if you have to talk about, serum is equal to plasma minus clotting factor. So they do not have clotting factor, but plasma is going to have clotting factor. This is going to be what? Clotting factor they do not have. So the answer is going to be option B. Okay. Next one. Name the blood cells which helps in reducing clotting factor, leading to excessive loss of blood from the body. So if there is no thrombocytes, platelets, ultimately it causes what? This one. Okay. Next one. Okay, we'll see this question. So adult human beings are enucleated. Yes, no. Why? What's the purpose? They are somatic cells. They do not need to reproduce. No. Why are they actually not having that? Because they need large amount of space. So their internal space is available to transport oxygen. They are not utilizers. They are actually transporters. So the answer is going to be this one. Yes, I'm going to give you one work for you. So you're going to solve the sixth question, which is going to be in a cardiac output. Cardiac output is given and heartbeat is given. You have to find out the stroke volume. So in the next class, I'm going to ask this question. So everybody is going to come and we are going to do this question. So I think I'm going to end the session. I hope everybody is liking the session. And I have talked about the human heart, blood, which are very important for your examinations. And this is very important. And we are also going to come on 15 days. And you'll be having a lot of classes from 4 to 5 a.m., which will be live. You can also ask questions. And I'm going to end the session for now. Thank you all of you for joining, and make sure believe yourself and you can do. If your victory is very close by, don't dare to actually give up, and all the very best for all of you. And I'm going to come in the next classes and meet you back again. Thank you all of you for joining. We'll meet in the next video.